DNA replication is going to occur during the S phase of the cell cycle. And remember, that is when the cell is going to be getting ready to divide. So the cell needs to replicate its DNA or its chromosomes in order to make sure that each of the daughter cells gets a copy of each of the chromosomes. So that's why the DNA is going to be replicated. So there are many enzymes that are involved in DNA replication, but the only one that you need to remember is that DNA polymerase makes the new DNA strand. So if you just remember that DNA polymerase makes DNA, hopefully that will stick and you can remember that. The two DNA strands are complementary to each other. And that just means that the A's and T's go together and C's and G's go together. So if you just remember apple tree and chewing gum, that's what complementary means. And so if you know the sequence of one strand, you'll be able to figure out the sequence of the other strands because they're complementary to each other. So let's just take a look at this DNA template strand down here at the bottom. And so we've got one strand running 3 to 5 prime. And remember, the other strand would always run in the opposite direction, so it would run 5 to 3 prime. So the new DNA strand we're going to be making is going to be complementary to the old strand. So if we have a T in the old strand, the new strand would have an A. And then if we've got a C in the old strand, we'd have a G in the new strand. And then if we have a G, we have a C, and an A would go with a T would go with an A, and then our last one would be a C. Okay, and then here's just another picture, just showing the double-stranded chromosome, our DNA molecule that we're going to be starting with, and our cell's getting ready to divide, so we need to replicate this chromosome to make sure that we end up with 2 of these chromosomes. So then we can have one chromosome going to one daughter cell and the other going to the other daughter cell when the cell divides. So what we're going to do is we're going to break the hydrogen bonds between the 2 DNA strands and separate the 2 old strands of DNA and use each one as a template, and then make a new strand to go with each of the old parent strands. And so now the one chromosome that we started with, we now have 2 copies of that chromosome. And so here are the 2 copies of the chromosome that are going to be attached together, and then we will separate these when we go through cell division. And those will end up going to the individual daughter cells, so each cell will have a copy of the chromosome.